مرحبا طلابنا الأعزاء. The title of this lecture today is Preformulation of a Drug. In this lecture, I'm going to discuss such important factor which have to be studied in formulations of a drug to make a dosage form comply with the international requirement. The most important properties and factor we have to study during the pre-formulation time is the physicochemical properties of the new drug that could affect the performance of a drug in its dosage form. Most of these information can be obtained from the previous study and research from published data or paper. Also, some experiment can be held by the pharmacist to get the informations of the solubility, nature of the substances, the crystals type and identity of the drug, polymorphism of the substances, and so on, of this characters or the property of the drug. Modification of a drug. In most cases, the new development drug do not meet the requirements of with the physicochemical properties. Therefore, we're going to make such modifications. Prior to starting a pre-formulation study, the physical pharmacist should meet with the principal investigator involved in the drug's development to obtain information on non-properties of the compound and the proposed development schedule. A salt formation of a drug has proved the best approach for development of dosage form. In general, the organic salt are more soluble than corresponding unionized molecule, that means the base, and hence increase the dissolutions. An example for that, we have ephedrine hydrochloride. Ephedrine as a base is less soluble than the salt, ephedrine hydrochloride, and that's why we use ephedrine hydrochloride in the preparation of nasal drops. Information for designing of drug dosage form. In fact, the formulation of new drugs or dosage form is similar or like the research work. It's need collection of different informations about the drug materials, including the identity of the drug, its structure, formula, and molecular weight, its melting point, crystallinity, and polymorphism, and these properties and the study of these properties can be held by different analytical method, like the HPLC method, UV visible spectrophotometer, TLC, mean the thin layer chromatography, and other type of the analytical method used for the to obtaining the information of the drug. Also, we have to collect a data of the therapeutic dose and the bioavailability of this drug. The other data is the bulk data, including the particle size, type of crystal, solubility, and bioavailability, and so on. Method of analysis for validation and stability study. We have to get a stability indicating method for the drug to study the degradation, because this type of method should be specific for the degradation, to identify the degradation of the drugs over the storage, so we can establish the stability information by following the drug degradation. These can be followed by using different methods like the HPLC, TLC, and UV visible spectrophotometer. Also, we need clinical test information, either from the research data or the index books, references, other references to give the limited dosage form and the dose for the child and the adult. Bulk characters of the drug. Bulk properties for solid material of the drug and the particle size, the bulk density, and surface morphology. All of these characters interfere with the procedure or the process, industrial process. Bulk properties for the solid form, such as particle size, bulk density, and surface morphology, are also likely to change during process development. Therefore, comprehensive characterization of all pre-formulation bulk load is necessary to avoid misleading prediction of stability or solubility, which depend on particular crystalline form. الحقيقة احنا بالرغم من انه هاي البروبرتي قد تكون للمادة الأولية للسوليد ماتيريال published، يعني معينة ومكتوبة، لكنه خلال البروسيجر العملية الاندستريال بروسيجر قد تتغير، ولهذا لازم نجري عدة تجارب نشوف شنو تأثيرات البروسيس الاندستريال بروسيس على الخواص الأساسية للمادة من ناحية الدنسيتي والكريستالينيتي، وأهم شيء morphology of the substance قد تتغير من مورف إلى مورف آخر. وهذا معروف أنه بعض الدراج مورفز polymorphs تكون أقل solubility، أقل bioavailability أو activity من مورف أخرى، فالتغير بها يؤثر على البيولوجيكال اكتيفيتي اوف دراج. فلازم احنا نجري عدة تجارب بالبري formulation to avoid these changing in the physicochemical properties of the drug. Crystallinity and polymorphism. Crystallinity of the substances and its behavior is so important in a drug formulation. Crystal habit and the internal structure of the drug can affect bulk and physical chemical properties of the drug, which range from flowability to chemical stability and so on. Habit of the crystal is the description of the outer appearance of a crystal, whereas the internal structure is the molecular arrangement within the solid. The other properties of the drug, like the hydrate property. Hydrate property should be identified. The hydrate compound is important since their aqueous solubility can be significantly less than their anhydrous forms. Conversion of anhydrous compound to hydrate within the dosage form may reduce the dissolution rate and extent of a drug absorption. بالحقيقة هاي مهمة جدا لعلاقتها بال plan of the industrial process for the dosage form of a definite substances. احنا بالبداية لازم نعرف كقاعدة ان الهايدريت substances يعني اللي بيها molecule of water, one or two molecule of water, its solubility less than the anhydrous. Therefore, we try to prepare or use anhydrous substances, which increase the solubility and therefore will increase the dissolution and the absorption of the drug. Polymorphism is another property which affect the stability and solubility of the drug. Polymorphism is the ability of compound to crystallize as more than one distinct crystalline species with different internal lattice. Chemical stability and solubility changes due to polymorphism can have an impact on the drug's bioavailability and its development program. Example of this effect on chloramphenicol palmitate, which exists in three crystalline polymorphs, polymorph A and B and C. Polymorph B has more solubility than other polymorph of chloramphenicol palmitate. Therefore, the serum level of the absorption increased substantially as a function of the percentage of form B polymorph. We continue with the polymorphism. polymorphism can be classified as one of two types. Enantiotropic, that means one polymorph can be reversible changed into another by varying temperature or pressure, as sulfur. And the other type, monotropic. This polymorphic form is unstable at all temperature and pressure as a glycerine stearate. We can hold this study about the enantiotropic and the polymorphism by using different method and instrument like microscopic method, fusion method, and differential scanning calorimetry, which is called DSC, and by infrared spectroscopy, X-ray powder diffraction, and scanning electron microscopy, dissolution and solubility analysis also. There is no general way of relating enantiotrope and monotropy to the properties of the polymorphs, except by locating the transition temperature or the lack of one. At a specified pressure, usually 1 atmosphere, and the temperature at which two polymorphs have identical free energies is the transition temperature. And at that temperature, both forms, mean the enantio and the monotropic, both of them forms can exist and have identical solubilities in any solvent as well as identical vapor pressure below the solid melting temperature. The polymorph with the lower free energy corresponding to the lower solubility or vapor pressure is the thermodynamically stable form. During the preformulation, it is important to identify the polymorph that is stable at room temperature and to determine whether polymorphic transition can be possible within the temperature range used for the stability study and during the processing, drying and milling. بالحقيقة ما دام industrial process like the stability study, during the stability study we subjected the product to the higher temperature as an accelerated temperature like the 40 degrees centigrade and humidity, and also during the drying process and the dosage form and milling. All of these will raise the temperature. So we have to select the stable polymorph of the drug with these process effect. Other solid properties. Hygroscopicity. Many drug substances, particularly water-soluble salt forms, have a tendency to absorb atmospheric moisture. Absorption and equilibrium moisture content can depend upon the atmospheric humidity, temperature, surface area, exposure, and the mechanism for the moisture uptake. Deliquescent material absorbs sufficient water to dissolve completely, as is observed with sodium chloride on a humid day. Other hygroscopic substances absorb water because of hydrate formation or specific site adsorption. With most hygroscopic material, changes in moisture level can greatly influence many important parameters such as chemical stability, flowability, and compactability of the powder. يعني نلاحظ هاي البروبرتي tendency to absorb water absorption of water. طبعا كلمة الادsorption تختلف عن الابsorption. Adsorption mean on the surface، يعني الماتيريال تاخذ المويستشر interfere with the surface of the particle called adsorption. هاي العملية راح تأثر على الويت of the substances. يعني إذا عندنا مادة امتصت 5% من moisture حسب وزنها، فإذا أريد أني أحتاج 100 ملغرام أخلي بالحباية من المادة، خلينا نقول بروفين 100 ملغرام. إذا هو ممتص ووتر فلازم مثلا بنسبة 5% لازم أوزن 105 حتى أحصل على 100. ولهذا هاي العملية لازم أسويها قبل ما أسوي الفورمولا، ولهذا هي واحدة من الدراسات اللي تطلق عليها preformulation study. قبل أن أسوي فورمة لازم عندي preformulation study عن المي، كمية المويستشر بالمادة. الفاين بارتيكلز لازم أسوي لها تحديد لتأثيرها على البالك ولا density وتأثيرها على dissolution also. Bulk flow formulation, homogeneity, and surface area control the process such as dissolution and chemical reactivity are directly affected by the particle size and shape and the surface of morphology of the drug particle. In general, each new drug candidate should be tested during preformulation with the smallest particle size, as is practically it facilitates the preparation of the homogeneous sample and maximize the drug's surface area for interaction. The particle size can be measured in solid by using a series of sieves, starting with the number 200 mesh for size 75 micron. There are other methods as scanning microscope and nitrogen adsorption BET method. The bulk density, these other properties can be studied before formulation. That's why it is one of the pre-formulation study. Bulk density of the compound varies substantially with the method of crystallization and milling or formulation. Once density problem is identified, it is often easily corrected by milling, slugging, or formulation. Usually, bulk density is of great importance when one considers the size of high-dose capsule or product or the homogeneity of low-dose formulation, in which there are large differences in drug and excipient density. احنا درسنا البالك density عمليا وشفنا انه الدنسيتي in bulk تختلف عن التابد when compressed، ولهذا بالكبسول فيلينج مهمة البالك density نعينها ونحسبها بالفيلينج مال الكبسول، لأنه الماشين تطلق بدون بريشر كمية من الباودر اللي يسد كل الكافيتي مال الكبسولة، البودي مال الكبسولة، فلازم نعرف ايش قد تتحمل هاي الكبسولة من البالك density، ايش قد حسب الفوليوم. وكذلك الاكسبيانت density همينة مهمة جدا بالحساب. Other properties, the powder flow properties. Pharmaceutical powders may be broadly classified as free-flowing or cohesive, non-free-flowing. Most flow properties are significantly affected by changes in particle size, density, shape, electrostatic charge, and adsorbed moisture, which may arise from processing or formulation. The flow property of solid powder and its percent of compressibility can be calculated by application of certain equation. These values are necessary in pre-formulations. هاي مثل ما تحقق عندنا عمليا حتى تكون يكون الباودر والفورمولا ready to flow in the industrial machine or compress. We have to study the powder flow properties by applications of the equation specified for this property. Solubility analysis. For oral dosage form, solubility is very important. Pre-formulation solubility study focus on a drug solvent system that could occur during the delivery of a drug candidate. For example, a drug for oral administration should be examined for solubility in media having isotonic chloride ion concentration and acidic pH. Even though the rules of administration may not defined at this time, understanding the drug solubility profile and possible solubilization mechanism provide a basis for later formulation work. Pre-formulation solubility studies usually include determination of pKa, temperature, dependence, pH solubility profile, solubility products, solubilization mechanism, and rate of dissolution. Analytical methods that are particularly useful for solubility measurements include HPLC, UV spectroscopy, fluorescence spectroscopy, and gas chromatography. For most drugs, reverse phase HPLC offers an efficient and accurate means of collecting solubility data. Solubility and dissolution experiments should have all factors defined, including pH, temperature, ionic strength, and buffer. For an equilibrium solubility determination, excess of a drug dispersed in solvent is agitated at constant temperature. Samples of the slurry are withdrawn as a function of time, clarified by centrifugation, and assayed to establish a plateau concentration. The solid precipitate is also characterized to establish the equilibrium solid form of the drug. بالحقيقة هاي الميثود of analysis of solubility احنا ماخذين انتو طلاب يمكن بالتكنولوجي ماخذين الاسبرين solubility شلون نعين solubility of aspirin وهي عبارة عن انه في حجم معين من الووتر لو فرضنا 100 مل نذوب كمية in excess of aspirin زيادة من الأسبرين كمية في temperature معينة ثابتة لو فرضنا room temperature 25 degree centigrade مع الاجيتيشن يعني التحريك المحلول راح يحدث الساتوريشن. ناخذ سامبل من المحلول ونسوي له سنترفيجيشن نحصل على كلير سوليوشن. هذا الكلير سوليوشن نسوي له اناليسيس. طرق الاناليسيس اما بال HPLC اللي هي بيس ميثود احسن الطرق، واحيانا بالبساطة باليوفي فيزيبل انسترومنت نحقق هاي النتيجة انه نستخرج كمية المادة اللي ذابت بالمحلول المعين، فنختار كمية معينة مثلا 10 مل ونسوي لها determination assay نشوف ايش قد ذائب بيها، فنعرف نطلع الكمية اللي ذابت بال100 مل اللي استخدمناها بالبداية. فمن هاي نعين solubility هي saturation time، هي الاكويليبريوم تحدث لما يصير saturated solution of the material and the ولهذا كذلك نحقق السوليبيليتي بوجود الايسوتونيك سوليوشن كمية يعني 0.9% والصوديوم كلورايد موجودة بالسوليوشن، واحنا نريد نذوب المادة بها. اما نستخرج هنا السوليبيليتي كمية السوليبيليتي من المادة من الدراج بوجود الصوديوم كلورايد، وكذلك من different temperature pH in acidic or basic pH كذلك نستخرج السوليبيليتي، لأن السوليبيليتي كذلك معتمدة على ال pH مال المحلول، أحيانا more soluble in acidic than in basic وهكذا. Solubilization. For poor water-soluble drug. Pre-formulation studies should include limited experiments to identify possible mechanisms for solubilization. A general means of increasing solubility is the addition of a co-solvent to the aqueous system. The solubility of poorly soluble non-electrolyte can often be improved by orders of magnitude with a suitable co-solvent, such as ethanol, propylene glycol, and glycerin. These co-solvents solubilize drug molecules by disrupting the hydrophobic interaction of water at the nonpolar solute by water interfaces. The extent of solubilization due to the addition of co-solvent depends on the chemical structure of a drug. That is, the more nonpolar the solute, the greater the solubilization achieved by co-solvent addition. For hydrocortisone, as example, hydrocortisone 21 heptane sulfonate, the lipophilic ester is solubilized to a greater extent by addition of propylene glycol than by more polar parent compounds. Some poorly soluble drugs can be solubilized in micellar solutions, such as 0.01 molar between 20, or by molecular complexes as with caffeine and cyclodextrin. These specific formulations can be tested with the addition of material on the solution medium as an experiment or pre-formulation experiments. Dissolution of the dosage form. Soon the product of the drug is manufactured. Dissolution should be studied to identify the bioavailability problem area. Dissolution can estimate the amount of drug which may reach the bloodstream and impact the drug biological activity. Therefore, dissolution is a competitive factor for the drug products between different company or producer. Dissolution can be controlled by different physicochemical properties like the particle size of the raw material, crystal form, and polymorphic forms of the drug, and the solubilizing agent we have used in the formula, and so on. Stability study. Pre-formulation stability study are usually the first quantitative assessment of chemical stability of a new drug. These studies include both solution and solid-state experiments under conditions typical for the handling, formulation, storage, and administration of a drug candidate. This section focuses on the evaluation of chemical stability during pre-formulation research. The first lot of the production can be subjected to the stability program either by the accelerated stability study or long-term stability study, upon which the result of this study indicate or give the permission for the using of the drug after a while. This study include the determination of the nature of degradation products. We have to study the degradation process of the compound, the chemical changes during the effect of storage at different temperature or the accelerated temperature. And the result of this study can be controlled by different analytical method like the HPLC indicating stability method. Estimation of expiration time of the products. As a result of the stability study, we can go to estimate by certain equation for the accelerated stability test or by long-term storage for 3 or 4 years storage and study. We can estimate the shelf life of the drug product by control or follow the changes in the percentage or the nature of the drug. If there is a degradation of products, significant amount or not. And this depends on the 10% of loss in the drug percentage.